Hello students, today the topic is genetic code. So what is this genetic code? Okay, so we know that all the genetic information is present on DNA and is also present on RNA. That we discussed already in transcription. Means what? The genetic information present on DNA is transcribed. Means what? The scripture is transferred in the form of messenger RNA. That is what is transcription means. So now this genetic information which is present on DNA and RNA should be expressed. And how it is expressed? It is expressed in the form of a protein. It is expressed in the form of a protein. Means what? After transcription, there will be translation, as a result of which protein is formed. And I think in the chapter you have studied about proteins. Proteins are not homopolymers, they are heteropolymers. That means they are made up of different types of amino acids. Means almost 20 different types of amino acids will make a protein. And in every protein, different types of amino acids will be there. If same amino acids will make all proteins, then all proteins will be of one type. So different types of proteins are formed because of the presence of different types of amino acids. So that means this information of number of amino acids, sequence of amino acids is present on DNA, and that information is transcribed onto mRNA, and this mRNA is carrying that information and is undergoing translation, and during which amino acids are arranged according to the information. And from this we can say that what is the genetic code? There is a interrelationship between, okay, the polynucleotide information present on DNA and RNA with okay, the sequence of arrangement of amino acids in the polypeptide chain is called genetic information. I hope don't get confused. Uh, let us make it clear. So, what is genetic code? It is the interrelationship between polynucleotide sequence, polynucleotide sequence present on DNA and RNA, and the sequence of amino acids in the polypeptide chain is called genetic code. That is what the introduction and the definition. And remember that cracking this genetic code or okay, proposing the genetic code and also deciphering the genetic code is a challenging task, and many disciplines are involved in deciphering the genetic code. What are those disciplines, or what are the branches of science or scientists involved here? Physics people, that is physicists, organic chemistry people, that is organic chemists, okay, biochemistry people, that is biochemists, and also genetics people, that is geneticists. geneticists. All of them are involved in cracking this genetic code. Okay, and now let us say the sequence of events that took place in the history. Okay, first we have to mention, so the name which is worth mentioning here is George Gamow. And he is not a biologist, he is physicist. And remember, this George Gamow has coined the term genetic code. The term genetic code was coined by George Gamow. And not only that, what he said, he made one argument, and knew that the RNA is composed of four nitrogen bases. Let us say one is uracil, second one is cytosine, third one is adenine, and the fourth one is guanine. So there are only four nitrogen bases, and if one nitrogen base is allotted to one amino acid, then it becomes a singlet codon. And then how many that means how many amino acids will get these codons? Only four. Now I hope we are understanding. In protein synthesis, 20 amino acids are participating. But here the singlet codon okay, justifies how many amino acids? Only four. So how many are not getting the codons now? 16. That's why some people they thought that he said even if the combination of these four, that is 4 square or 4 into 4 or 4 square, will give you how many codons? 16 codons. So even this can be called as what doublet codon, and this doublet codon is also not sufficient, because only 16 amino acids are getting the codons and the 4 are not getting. So this is also insufficient. That's why at this moment, George Gamow has made a bold proposition. He said that it should be 4 into 4 into 4, that is 4 cube, and that results in the formation of how many codons? 64 codons. So actually remember that, is it sufficient or insufficient or more than sufficient? It is more than sufficient, because there are only 20 amino acids and the number of codons that we are getting are 64. More than sufficient. And now so again, so okay, the confirmation of this has been started again. And among that, many scientists, among which I wrote some of the names here, which you have to memorize: Marshall Warren Nirenberg and J. Heinrich Mattei, Hargovind Khorana or H.G. Khorana, and Severo Ochoa. These people, okay, and they have done some experiments to find out whether it is doublet codon or singlet codon or triplet codon is working. Okay, and these Marshall Nirenberg, Marshall Nirenberg, and Mattei, they have okay, they made one poly-urna, Is what a synthetic RNA they have prepared, and that is what poly-U RNA. What does it mean by poly-U RNA? That is what it is, a RNA composed of only uracil, U. And now I am just dividing this into 3, U. Okay. So, how many triplet codons here? 1, 2, 3, 4, 5. Of course, just for our understanding, I divided it, but they were not aware of this. And when they have added this poly-U RNA in a medium where they have supplied tRNA and amino acids, all 20 amino acids are there. And they got one polypeptide made up of only phenylalanine. Phenylalanine. I am writing phe. Okay, you can see that how many phenylalanine chain is formed? 5. So that indicated that it is 3 letters or 3 nucleotides of RNA are coding for one amino acid. And this is the first codon discovered. What is that first codon discovered? U. And it codes for which amino acid? Phenylalanine. Okay. Then remember only one they discovered, and Hargobind Khorana, and he prepared a synthetic RNA from the DNA, and he made homopolymers RNA and copolymer RNA. Means what homopolymer RNA means? Only one type of nitrogen bases or nucleotides. Copolymer means what? Two types of nucleotide chain. I hope you understand. The homopolymer RNA is made up of single type of nucleotides, whereas copolymer RNA is made up of what? Okay, so two types of nucleotides. And for example sake, let us take one RNA. Okay, prepared by Hargobind Khorana or synthesized by Hargobind Khorana. Cu, 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 Cu. Okay, and I am dividing this. 1, 2, 3, 4, 5, 6. So 6 triplet codons are there now, and it is made up of only 2 nucleotides. That's why it is a copolymer RNA. That is what cytosine and uracil. Okay, and what has happened? You see, this CUC translated into lysine, and UCU is translated into serine. Okay, lysine, serine, lysine, serine. So 3 lysines and 3 serines are formed. You can see like this. So that means now Hargobind Khorana discovered the codon, 2 codons. One is CUC, the other one is UCU. CUC and UCU. And that Marshall Nirenberg and Mathieu, they discovered what? UUU. For which one? Phenylalanine. And the CUC for lysine and UCU for serine. Okay, and you see, only three codons for three different amino acids were so far discovered. And then what about the remaining 17? So the remaining 17 codons are discovered because of a Spain scientist, Severo Ochoa. And he made one enzyme called polynucleotide phosphorylase enzyme. He discovered this enzyme. What is that enzyme? Polynucleotide phosphorylase. And it is also popularly known after the scientist as was Severo Ochoa enzyme. Severo Ochoa enzyme. I hope you understanding. In the examination, if they ask which one among the following is Severo Ochoa enzyme, you have to go for this polynucleotide phosphorylase. And the significance of this enzyme is that it will polymerize a reaction independently without a template. It is template independent RNA polymerase enzyme. Don't forget that RNA we already discussed in the transcription, and the transcription was catalyzed by the enzyme RNA polymerase, which is DNA dependent RNA polymerase, because it can't polymerize or it can't catalyze the reaction of polymerization without the template. But this enzyme will synthesize RNA without the template. So this is called enzymatic synthesis of RNA. Okay, I hope understanding. Enzymatic synthesis of RNA is possible with which enzyme? Polynucleotide phosphorylase enzyme or Severo Ochoa enzyme. And because of this, they, with the help of this enzyme, they have created different RNAs with different combinations of nucleotides. And when they have added those different types of RNAs in the medium containing different amino acids, they have got different types of proteins with different types of amino acids. Thus, finally, the scientists have succeeded in deciphering the genetic code, and they have created a checkerboard for this genetic code, which I drew on the board here. Okay. And even that now, I hope you are understanding which codon is now confirmed. Is it singlet codon or doublet codon or triplet codon? Triplet codon. What is triplet codon? So the triplet codon is composed of three successive nucleotides, not three alternate nucleotides. Three successive nucleotides present on RNA will behave as a code or a triplet codon. Now, here you see in the checkerboard, you see I wrote UCAG, that is what the first letter. This is first letter. And here also again I wrote in the vertical columns as what UCAG. So that is second letter. And now for each horizontal column, I wrote UCAG here, UCAG, UCAG, UCAG. That is what third letter. So this is second letter in the codon, and this is third letter in the codon. And now I hope filling all these okay boxes. How many boxes? Okay, 16 boxes can be filled, and each box will get how many combinations of codons? 4 combinations you can see here. Okay, first letter U, second letter U, and the third letter is also U. Means what? Third first letter is U. And like that, first letter, second letter, third second letter here C. Okay, first letter, second letter, and third letter here is what? A, and the first letter, second letter, and the third letter here is G. Okay, and actually while filling it, you had to write first these two, U, and then you write simply UCH. Okay, like that, U and second letter C, UC, you write 4 times and then UCH. Like that, you fill all these and practice it, and remember these amino acids. So UUU codes for phenylalanine, UUC codes for phenylalanine, UUA leucine, UUG leucine, like that. You just memorize these things. But among these 64 codons, okay, how many are actually coding for amino acids? 61. 61, and these 61 codons are called sense codons. And the three remaining codons, you just see here, I made a box here, and UAA, UAG, and UGA, and there I wrote stop. They are stop signals. That is what the three are called. Okay, nonsense codons or terminating codons or stop signals. Stop signals. So that is what about the genetic code, and now we shall discuss about the salient features of genetic code. Okay, now let us discuss the salient features of genetic code or properties of genetic code. The first one is what the genetic code is a triplet codon. The three successive nucleotides, okay, the three successive nucleotides will behave as a codon or triplet code. Okay, all right. And it is non-ambiguous or specific. Means what? One codon specifies only one amino acid. That means always that codon, okay, uh, okay, the codon, okay, codes only one amino acid. That is what specificity. So that's why there won't be any ambiguity here. So that's why it is called non-ambiguous. And doing that, these codons are all arranged contiguously on mRNA. The codons are all present contiguously on mRNA, and we should not expect any punctuation marks here. There won't be any punctuation marks. So that's why it is comma-less. And degeneracy of code means what? Actually, there are 61 codons and there are only 20 amino acids. That's why some amino acids got not one codon, they got what? More than one codon. For example, you just look at this leucine. Leucine got 6 codons, and you can see serine. Serine is also having 4 here and 2 here, so total 6. And also like that, you can see arginine, 4 here and 2 here, so 6. And some got 4 codons, that's why. So this is called degeneracy. Means what? Codons are different, but the which amino acid is coming, same amino acid is coming. But to this, some exceptions are there, like methionine and tryptophan. Methionine and tryptophan. Okay. So the methionine is always coded by AUG, and tryptophan is coded only by one, that is what UGG. So that is what exceptions. So that is what, okay, degeneracy codon and universal code. Is what this is applicable to not particular species or not particular organism. It is applicable to all cellular organisms. But to this property also we find some exceptions in the mitochondria. In the mitochondria and in some protozoans, there is some exceptions will be there. Okay, and now let us see the sixth one, that is initiation codon. That means this translation always starts with a particular codon, and that is called initiation codon, and that initiation codon is AUG. And this AUG codes for methionine, and this AUG is playing one dual role here. Whenever it is present in the beginning, it behaves as initiation codon and codes for methionine only. But that doesn't mean that this AUG is present only at the beginning. It may be present even in the between, and even when it is in between, it codes for methionine only. That is what dual role. It behaves as a initiation codon as well as codon of methionine. I hope you are understanding this point. And the next one, stop signals. That means this mRNA, and you see it will have 5 prime and 3 prime ends. Okay, and you see at the 5 prime end you will find the initiation codon and many other codons, and let us say at the end you will find one codon, okay, which is called stop signal or terminating codon. And this can be UAA, or this can be UAG, or it can be UGA. Okay, don't think that all three are present at this 3 prime end of RNA. Any of these three is present at the end of RNA. Okay, so that is what terminating, and just remember they are given some names also. UA is popularly called as Walker, and UAG is called Amber, and UGA is called as Opal. Okay, UAA Walker, UAG Amber, and UGA Opal. And the last but not the least is what non-overlapping. And what is this non-overlapping? One codon will not overlap on the next codon. Okay, they are present independently and they code the amino acid independently. Okay, so that is what is about the topic genetic code.